Globin genes had evolved long before vertebrates. And by the time the first vertebrates had evolved, already some duplications had occurred in globin genes, and so that the ancestral vertebrates had multiple globins. Very old members of the globin family tree include cytoglobin, which is located on human chromosome 17 and expressed in many cells, and neuroglobin, which is located on human chromosome 14 and is expressed in the brain. Also around this time, there must have been the ancestor of both myoglobin and hemoglobin. Prior to the evolution of the jawed vertebrates, this final globin had been duplicated, and one copy will ultimately become the myoglobin, located on human chromosome 22, which is expressed in muscle, and hemoglobin. This early hemoglobin gene was subsequently duplicated to form the alpha hemoglobin ancestor and the beta hemoglobin ancestor. The alpha hemoglobin ancestor, located on human chromosome 16, was then duplicated multiple times to produce a cluster of alpha hemoglobin genes, which include the zeta hemoglobin, the zeta pseudogene, the alpha pseudogene, the alpha 2 gene, the alpha 1 gene, and the theta gene. The beta hemoglobin ancestor on chromosome 11 in humans was subsequently duplicated to form the epsilon globin, the gamma G globin, the gamma A globin, the eta globin, the delta globin, and the beta hemoglobin. So all of these globins in the human genome form a gene family. They are descendants from ancestral globin genes which underwent duplication and then subsequent modification.